Welcome everyone to today's Biological Control Workshop webinar. We're so excited to have you all here from across Australia and to learn about all things biocontrol. For those who don't know me, my name is Beck James and I'm the Natural Environment Program Officer for the Department of Environment, Land, Water and Planning, or DELWP, and I'm based in Bendigo in North Central Victoria. I'll be your MC for today, and I'll also have a few helpers today as well to make sure that it's a smooth experience for everyone. We have the Weeds at the Early Stage of Invasion team from DELWP, Bianca Gold and Kate Blood, who are helping us with the tech side of things, and Kate will be assisting at question time as well at the end of the presentations. So I'd like to begin by recognising that I'm on Dja Dja Wurrung country, and I pay my respects to the elders, past, present and emerging. However, I recognise that we are meeting from locations across Australia, so we will be on the lands of other traditional owners, and we thank them for their custodianship of the land. I'd also like to welcome any Aboriginal elders who may be joining us today in the workshop. So let's get started. So we have an action-packed lineup for today. We have Dr. Raelene Kwong and Greg Lefoe, who are senior research scientists from Agriculture Victoria's Research Division, to explain all the ins and outs of what's involved with biocontrol and how you can get involved in your patch. We also have another special guest, Mead, who's the president of the Tarrangower Cactus Control Group, who will provide an exciting case study around the use of biological control for wheel cactus or Opuntia robusta in North Central Victoria. So the DELWP Natural Environment Programs team is proud to host the Loddon Mallee event for today and for the Connecting Communities series. And this event is generously supported by DELWP's Weeds at the Early Stage of Invasion project, Connecting Country and Weed Society of Victoria. We also have an additional event sort of tacked on to the end of this as well at 11:30, where you're welcome to join the Weed Society of Victoria's annual general meeting, which goes to about 12. So without further ado, we'll jump into it. So before I just jump in, there's going to be two presentations in this block. So we'll kick off with Dr. Raelene Kwong, who is a senior research scientist in Agriculture Victoria's Research Division. Ray leads Victoria's research and development programs on the biological control of weeds and invertebrate pests. With more than 15 years experience in the field of biological control and studying a PhD in entomology with La Trobe Uni, Ray now oversees a host range testing candidate agents against several weed species. She's here to talk about them today and teach us all things biocontrol. Over to you, Ray.
Perfect, thank you. And geez, it's so exciting to see so many people interested in biological control of weeds. And I wondered whether all of this recent interest has something to do with this beautiful plant that is just gracing our landscape at the moment, and that is Patterson's curse. I certainly have been getting a lot of inquiries about Patterson's curse, particularly about biocontrol in recent weeks. And so some of these questions I've been getting are things like, the biocontrols have stopped working. Has the Patterson's curse become resistant? And where have the weevils gone and how do I get them back? And so with all of these questions, I started to think maybe it was time that we revisit some of the fundamentals of biocontrol. And so my presentation is really a bit more of an overview of biocontrol. And I thought it would be worth doing this because we've got such, I think, a diverse audience. There's like 250 participants here. So it's hard to kind of gauge where everybody is in terms of their knowledge of biocontrol. So let's just get back to some of the basics. Okay, so if we talk about the weed invasions and how biocontrol fits in, we would start with an exotic weed, coming into the country. And often when these weeds or when these plants are brought into Australia, they come in usually without their enemies, their natural enemies that would normally keep them in check within their native range. And so their populations can explode up into, these damaging kind of levels. And they naturally, though, these weed populations will fluctuate from season to season. But when we introduce biological control, our aim then is for the weed population densities to decline as the biocontrol agent populations increase. Now, obviously, this can take quite a fair bit of time for these biocontrol agents to increase to reach those, that damaging level. And so it takes, it's quite a long and involved kind of process. And this period here, could even be decades long. But once established, we hope that the weed population densities then become a lot lower. And certainly we hope that they're below that economic or that environmental damaged threshold level. And so this is what we're really aiming to achieve with biological control. But biological control will never eradicate the weed. And that's obviously important to know. And that's often one of the myths of biocontrol, and that is that will the agents eat all of the weed, and then what will they eat once the weed's gone? Well, biocontrol will never eradicate the weed. And so if we get back to our Patterson's curse story, and why is it that we're seeing lots of Patterson's curse around now? Well, Patterson's curse prior to biocontrol used to, obviously grace our landscape. And when the biocontrols were introduced, now the biocontrol program was, went for about 30 years. So it was one of our very long programs, started in about the 80s. There was about 7 biocontrol agents that were introduced, 6 of which are listed along the bottom here, are the ones that have become established. And so we have here a taproot flea beetle, a taproot weevil. This is a crown boring weevil. This is a stem boring beetle. This is a taproot flea beetle. This one is a pollen beetle, and this last one is a leaf mining moth. And these are all the ones that are established. And so this is one of our very successful biocontrol projects. And this is a photo of taken obviously at the same site in 2018, showing just how well those biocontrol agents had established. And it was really through the efforts during the 2000s, where through the Weed CRC and state agencies, across Australia, there was a massive concerted community engagement program in the rearing, release and redistribution of biocontrol agents for Patterson's curse right across the country. And that's why we're seeing such a great success. But why is Patterson's curse becoming a problem again? Now, so one of the issues, or there's a number of, things that can cause the weeds to come back. And one of them is the drought. One of them is overgrazing. It could be that when there's a late autumn break, all of these things can impact on the population of the biocontrol agents. And I think that this little weevil here, the crown boring weevil, has a really interesting kind of story in that what happens is that this little weevil over summer, the adults over summer go into a type of hibernation period when the Patterson's curse has died down. So they're kind of sitting in the soil. If there's a lot of overgrazing, a lot of trampling of stock, that can impact on the survival of those weevils. But the ones that do survive, they will come out of their summer hibernation, we call it estivation, in response to shortening day lengths in autumn. So there's a particular day length that they will come out. And when they come out, they're looking for new rosettes that have germinated following the autumn rains. If there's a delay in the autumn break, those weevils will emerge and there'll be no food for them, and then their populations will crash. And this is what we've seen, in the last couple of years due to the drought and due to late autumn breaks. And so our populations of insects, our biocontrol agents, have crashed. And then we've seen a resurgence in Patterson's curse, obviously with bare ground following fires and whatnot. And that's why we're seeing that resurgence. But that's not to say that the biocontrol agents aren't coming back. And this is just a photo I took down in Melbourne near the Holden Flora Reserve of the little grub, one of these little weevil grubs, working away in the Patterson's curse rosette. You can tell by all of this black goo. the biocontrol agents are there. They just need that little bit of time to build up. So don't despair. And it comes down to, that part of the last part of the invasion, curve and showing how biocontrol reduces the population densities. We're just going through, that cycle at the moment where the weed populations increase and then the biocontrol agent populations will increase and drop it down again. So just got to give them some time. But what I'm going to talk about now is then the actual process that we go through in a biocontrol program, because this is quite a long and involved process. Each individual stage of what we call the discovery to delivery classical biocontrol pipeline can take, 3 to 4 to 5 years to get through each of the stages. It could take 10, sometimes 15 years, depending on the complexity of the biocontrol program, to get to a point of biocontrol agents being ready for release. So I'm just going to take you through the stages that we go through. So first of all, we need to understand about the weed and what is it about the weed that makes it so invasive. And are there, parts of the weed's life cycle or parts of, its biology that we could target for biocontrol? It might be the Achilles heel of that weed. We also need to know things like why is it that the weed has become a problem in the invaded country? Is it something, is it that the weed has, evolved, or is it a different genetic entity, and all of these things? So these are some of the things that we study right at the beginning, and we compare our weed populations in the native range, where the weed originated from, to the invaded range in Australia, and see if we can understand what are the things about that weed that we could target for biocontrol. We then go through a process of finding out then what are the potential biocontrol agents that we could import. And we often say, well, I often say, it's like searching for the Holy Grail, because in the native range, that plant, that weed, it could have over 100 different natural enemies feeding on it and attacking it. Obviously, we can't import all of those. That would just be too risky a process. We just need to work out which of those natural enemies are going to be the best biocontrol agents. So here's our team of scientists over in Europe searching for our natural enemies. And when we have identified those natural enemies, then we need to look at, well, basically we want them to, fulfill 3 main criteria. So they need to obviously have an impact upon their target species, the target weed. They need to be able to establish in the introduced environment. And a lot of biocontrol agents that have failed in the past is because they just didn't establish in the introduced, when they were introduced into Australia. And there are a number of reasons why they may not have established. And it could just be the climate was not right for them. It could be that the weed population is a genetic entity from what they were collected from in the native range. So there are lots of different things that we need to look for. A lot of research goes into making sure that the biocontrol agents, those natural enemies, are going to establish. And obviously, the most important is that they need to have a restricted host range. So if you can imagine, you've got your suite of natural enemies. Some of those will be what we call polyphagous. They eat a whole range of different plant species, and some of them will be monophagous. So they're the ones that have co-evolved with that plant species, and they only feed on that. That's all they feed on that species, or maybe some very closely related ones. And they're the ones that we need to look for biocontrol. So how do we go about doing that? Well, that's when we need to go through a process of host specificity testing. And I've used an example here of silverleaf nightshade and a little leaf beetle that was under consideration as a biocontrol agent. Now, silverleaf nightshade is from the family Solanaceae, so that's related to tomatoes, related to eggplants. And we undertake these different trials to see whether the biocontrol agent, the candidate biocontrol agent, is likely to feed, lay its eggs, those eggs hatch and those larvae develop through back to adults. And these are the tests that we're looking to see whether that candidate biocontrol agent has a broad host range, is going to feed on other species, or is specific to our target weed. Now, how do we decide what plant species we should test our candidate biocontrol agents against? Well, we go through a process of selecting the species based on their relatedness to the target weed, and we use phylogenetic trees. So this is just an example, a generic kind of picture that I pulled off the web of a phylogenetic tree. And if we imagine that this is our weed, then the species that are closely related to it, these are the ones that are more likely to be attacked by the biocontrol agent if the biocontrol agent has a broader host range. So we're going to see feeding or damage or overposition on these closely related species first, and then maybe these species. But the chance of them feeding or living or accepting other plant species that are unrelated is very rare if the biocontrol agent is host specific. And so obviously in our testing, we can't test every species on Earth. It's just don't have the time or the resources. And so we have to be realistic, and we have to choose species that we think are going to give us, show us the level of that host range of that biocontrol agent. And so these are the types of species that we would select to undergo our host specificity testing. Now, depending on the weed, it might just be a handful of species if there's not that many species present in Australia. Or for something like silverleaf nightshade, Solanaceae, which has lots of different crop species, lots of native Solanaceae species, the list of species that would be tested is quite large, and that project, that testing process could take quite a lot of time. So we do this type of testing in a quarantine facility because obviously these insects or these biocontrol agents that we want to test are exotic, and we need to prove that they're safe for release. Now, I haven't really mentioned that a biocontrol agent can be an insect, it could be a mite, it could be a pathogen such as a rust. These are all different types of biocontrol agents that could be considered for use against a weed. And so this is just an example. This is our quarantine facilities based at AgriBio down in Melbourne at the La Trobe University facilities, La Trobe University campus in Bundoora. This is just an aerial view of AgriBio, and our quarantine facility is just based in this section here. These are our shade houses and glass houses. And so we need to propagate a lot of plants. This is our Sagittaria program, which is an aquatic weed, and all of these other tubs are where we're rearing or propagating our test species. These are the ones that will undergo the testing. They get brought into our quarantine labs where we've got controlled environment rooms and also glass houses, and we expose the biocontrol agent to each of the test species. This is just some of the staff and some of the students that have worked in our lab. And so when we undertake, once we've completed all of the those experiments, we then go through this risk assessment process. Now, this is a very complicated diagram, and I'm not going to go through it, but basically it is a way of showing you that our biocontrol agent goes through a series of different types of tests depending on the results of each individual test that they're exposed to. And so the actual testing process can be quite complicated, and we end up with quite a lot of data. All of that data then is put together into a submission document that then is submitted to the Department of Ag and Water, Water and Environment for a risk analysis. So DOR then take all that data. They do their own risk analysis report based on their import risk analysis process. And then they make a recommendation on whether they support the information in our application for release, whether they believe that the biocontrol agent poses, little risk to for release. That document then is circulated to all states and territories for review by, agencies in each of those areas, and also what used to be the Department of Environment, I'm not sure, it's now part of Department of Ag. It's also reviewed under a separate process, which is the Environment and Biodiversity Conservation Act. So the analysis process, or this risk analysis or this risk assessment process, is quite long and involved. It can take up to 2 years to go through this consultation process. There's also a period of public consultation where the public and industry are also able to scrutinise the results and say whether they support the release. When we do get the release approved, then we celebrate. Because it could have taken up to 10 years to get to this actual point of where we actually now have a biocontrol agent available for release. So I'm just going to quickly move on because I'm over my 15 minutes, and I just want to briefly talk about some of the projects. Now, we don't have time to go through each individual project, and we could have individual seminars on each individual project in the future if anybody is interested. But these are just some of the ones that we've recently been that we're recently working on. This is Sagittaria and Arrowhead, some aquatic weeds. We have 3 biocontrol agents that are undergoing testing, one of which has just been approved for release. And that's work that Jackie Steele and Uma Lubanga and myself have been working on. Silverleaf Nightshade is Greg Lefoe's project, and he's working on various new agents that will come into quarantine in the future. We have a new project starting this year on blackberry, looking at a cane boring sawfly, but also doing some additional native range surveys in the UK for some other potential agents as well. We have another new project. This is on serrated tussock, a project that we're running jointly with our New Zealand collaborators from Landcare Research New Zealand, where we're looking for some pathogens that have caused this dieback disease in Australia. It causes a fungus type disease, and we want to know what is this disease and whether there's any potential for development of that further. And there is also some work being done over in Argentina looking for some new biocontrol agents as well. Some of this work is funded by the Victorian Serrated Tussock Working Party. And Wandering Tradescantia, this is a bit of a project we're not currently working on at the moment because we've just completed the testing, the host specificity testing of this little leaf beetle here, and we will be compiling an application for its release for submission to the Department of Ag. So I'm going to leave it there. I've taken you through the discovery to delivery pipeline in terms of the research and development phase. Greg Lefoe is then going to talk about what I think is also a very exciting phase, and that is when we have biocontrol agents for release, and then how do we get them out there and how do you guys get involved in that process. So thank you very much. I think we're going to leave questions until the end of Greg's presentation.
Thank you. That was awesome.
Thank you.
So let's introduce Greg. So Greg Lefoe is the immediate past president of the Weed Society of Victoria and is a senior research scientist in the invertebrate and weed sciences in AgVic's research division. Greg has more than 25 years of experience researching invasive species management, especially the classical biological control of weeds and pest insects. Greg works closely with the Australian and international partner agencies, as well as producers, public land managers, and community groups to address pest and weed impacts on the environment, forestry, and agriculture. Thanks, Greg. Go for it.
Thanks, Beck. Great introduction there. And thanks, Ray, for that really good introduction to the next phase of biocontrol, which is implementing weed biocontrol on ground. And so, as Ray mentioned, this talk is at this end of the pipeline. So many years of research can go into a project before we get to this phase, but this is the phase where we have approval to release an agent, and we want to get it out there in the field and make it a useful weed management tool. What we've learned over many years is that scientists, when they're working with farmers, public land managers and community groups, can conduct many more biocontrol agent releases across wider areas than could otherwise be achieved. And this accelerates the spread of the agents and brings forward the benefits of biocontrol. So you can imagine a few releases, you might have effective biocontrol within 30 to 40 years. But if you can get many more releases across the landscape, that could be accelerated. So you're seeing control decades earlier. It's also important for ensuring biocontrol is integrated with property, local and regional weed management planning, because biocontrol is just one weed management tool. It's something that scientists just don't go out and release willy-nilly. It really needs to work in with local weed management planning to be effective. And so here are some examples of some agents that have been released in recent years or are actively being redistributed at the moment. The gorse thrips is one example which has been released in Victoria. It's very slow to disperse, and we're only seeing very low numbers at the moment. Gorse soft shoot moth is the most recent biocontrol agent released for gorse. And of course, gorse is a problem right across southeastern Australia. And its larvae chew on the soft shoot tips. We're in the process where we've conducted releases in South Australia, Victoria and New South Wales, and we're trying to get those agent populations established before we can harvest them and take them to other locations. But already in Tasmania, we're seeing really good results. Tasmania is a bit of a gorse haven, and often when we release biocontrol agents for the first time on gorse and broom, they do really well in Tasmania. And so we have an opportunity to collect from Tasmania and release them to other states, which is what we've done. English or Scotch broom, as it's known in New South Wales, is another target agent where we're actively redistributing agents. And there's an example there of broom gallmite. And this was Tasmania. You can see the broom is heavily galled, and that's depleting the plant's resources and in some cases killing large broom bushes. And we're starting to see results like that now on the mainland. Certainly up in northeastern Victoria, around places like Dartmouth, we're seeing good results with the broom gallmite. It's a really easy agent to redistribute. You simply need to take a cutting and place those galls on a new bush, and the mites will transfer over. So it's a really good one for redistribution by volunteer groups. But we do have barriers to effective engagement and implementation of biocontrol, and anybody who's tried to learn about biocontrol and implement it in their local area will understand what I'm talking about. So the barriers really are timely, ongoing and ready access to information on which weeds have been targeted for biocontrol, what agents are available for each of those weeds, how do you find the agents, and how do you collect them and implement biocontrol in areas where there are gaps. Fortunately, there are tools that can help us to address implementation barriers. There aren't as many on-ground weed officers these days who can assist with biocontrol directly, and funding is scarcer than it used to be. But platforms such as the Atlas of Living Australia, which is a huge database of plants, animals, insects and so on, is readily available to anyone to use and to adapt to their own purposes. And that's what we've done for weed biocontrol. In conjunction with the Atlas of Living Australia, we've built a biocontrol hub. So it's just a customised portal that allows us to upload biocontrol agent data, including citizen science data, and share that with the people who are working with weeds on ground. It also gives us free access to all of the tools and capabilities of the Atlas of Living Australia, such as their spatial tools. So if you Google the Biocontrol Hub and go to the front page, you can click on it and you'll find a list of 28 target weeds and their agents. And so here are some examples, so docks, English broom, gorse, biocontrol, and so on. So clicking on any of these will take you to the target weed. Here's English broom, for example. It comes up with the front page with lots of information about English broom, the agents that are available, and how you can participate in biocontrol of English broom. There are links and resources, so you can go to the Weeds of National Significance page, and you can then access all the resources on that page. There's a description of each of the broom biocontrol agents with links to forms where you can upload your observations or produce distribution maps and so on, and I can show you what they look like. So here's just a field that you can enter data into. So if you're monitoring sites or you notice a biocontrol agent out in the field, you can record it and it's uploaded and shared. And that's what we really like to encourage, because it gives us a lot more information about the spread and impact of biocontrol agents, but it also allows land managers to see where these biocontrol agents are, whether they're occurring in their area, and if they're not, where they can collect them from. You can produce distribution maps, and clicking on any of those dots can give you detailed information about that particular record and whether you can access it. Most of this capability has also been reproduced in a phone app, which is a really good achievement, which was funded by Agriculture Victoria, Meat and Livestock Australia and the Australian Government. And you can have this on your smartphone when you're out in the field. It produces all the same target projects. You can bring up a list of the agents. You can record sightings in the field and look at distribution maps. And that's available free of charge from Google Play and the App Store. So what does all of this mean? Well, using tools like this and the information and the power that we can have on our smartphone, we can access biocontrol information more easily, more readily. We can share information, and this can lead to really effective on-ground collaboration. In this top example, there's Sally Lambourne from DELWP at a site that is in the Central Highlands Eden project area, where she's releasing a biocontrol agent from gorse. Now, Sally was able to access information about the establishment of gorse offshoot moth from Tasmania. John Orison in Tasmania collected some larvae and shipped them over to Sally, and Sally released them in Victoria. And she has a tent set up to protect them temporarily. And Sally also recorded her release, so others can benefit from that site if it becomes established. Similarly, land care networks can work in collaboration as well. And here's an example from Beechworth, where I'm with Trevor Danger, the local land care facilitator, and we're inspecting a really well-established broom gormite site in Beechworth. And from that site, the Benambra Dinner Plain Omeo Land Care Group was able to acquire some galls and release them at a site just outside Omeo. There's the local grazier there, that's on her property. She's been really good at controlling broom, but this is a difficult to access gully where other control measures weren't possible. And so she's released broom gormite at that site to suppress the broom and prevent reinfestation to other parts of her property. So it's a really good example of how land care groups, using the data on the Biocontrol Hub, can collaborate with each other and share biocontrol agents. And here's an example of a better resource program where agencies from 3 states went down to Tasmania, collected gorse offshoot moth adults, and then took them to their individual states and conducted releases, and all of that data is on the Biocontrol Hub and is shared. And thank you. So just finally, acknowledgements. The funding support was really important for developing this tool, because we did recognise there is a serious gap in materials and information and data, and we've been able to address that somewhat, and we would encourage you to download the app or Google the website and participate. Thank you.
Thanks, Greg. That was awesome. Kate, over to you with the questions.
Wow, fantastic presentations. Thank you both. There's a couple of questions. The first one for Ray. Are there any updates on Gasterophyllus tricostalis for fireweed, and has the bushfires affected this research proposal?
That is a very tricky question. Gasterophyllus, I don't know it, but it is a stem-boring weevil for fireweed is all I know. I think CSIRO are working on this, and I'm not even sure, but please correct me if I'm wrong, because whoever, Mel, if you've asked this question, you might know more about it than me. My understanding was that this work was still in development, and that the South Africans were involved in this project and maybe doing some of the testing over there.
Yeah, as far
is that
as
right?
Yeah, as far as I know, they were doing some work in 2019, and on the 17th, they just got another, I think, almost 300,000 to work with this, specifically on the stem-boring weevil.
Yep.
And I was just looking at it. The fireweed is one of our big threats. Thankfully, we don't have it here, but it's spreading rampantly throughout New South Wales in a lot of places.
Mm-hmm.
I was just wondering if there was an update, because I know a lot of the areas in Big and down where they were doing testing got seriously affected.
Mm-hmm.
Was the money that came in and the approval in September, was that to reestablish and start over, or is that just an advance on and maybe we can start getting this thing out because it's got such a narrow host susceptibility?
Yeah, I think my understanding, and perhaps you're better off talking to CSIRO about this because that is their project. We've not had any involvement in this project. We don't have fireweed as one of our biocontrol targets for Victoria. But my understanding was that there was still testing to be done. So if they've received funding for that, then that would have been to continue with the testing. But yeah, I don't know. I don't have any personal involvement in that project. The same, I think you've got a question here on Parthenium wheat.
Yeah, same type of question. They were doing the Parthenium weed trials and testing, and it also has a very narrow host range.
Yep.
But Australia had a input into the Pakistani research that's going on, and Australia said it's got a narrow host weed. Is there a chance that we're going to be seeing that soon?
Well, I hope so, but I haven't seen any applications for release for either of those. So it just may be that they're still completing that work.
Yeah.
But it would have to go through, obviously, it would have to go through that testing process before we know. Yeah.
And Patrick's just typed in my third question, so I won't ask that one.
Okay.
Next question for Ray is testing on narrow host range on Listrasotus seticipennis in Australia. Based on what is currently going on in Pakistan, any Australian movement for Parthenium wheat, or is that what you just answered?
That's what we were just talking about, Kate. Sorry.
Beauty, that's covered. All right,
Yeah.
Next question for Greg. Where can we get the gall mite for broom? In the Riverina, Snowy Valleys and Murray areas, it is a big problem, likely to return with a passion after the bushfires.
Yeah, my advice is to go to the Biocontrol hub and just bring up a distribution map for broom gall mite. It'll have locations in New South Wales where broom gall mite is established. Certainly, it's an agent that readily establishes, so it's now widespread throughout northeastern Victoria, so that may be an option for some parts of southern New South Wales. But certainly if you go to the Biocontrol hub, there are resources there that'll tell you where it's established and how to collect it. Collecting it's really, really easy. You just need an esky and some pruners, and that's it. And if you can get in contact with local groups, they might be able to assist, or you can just drive there on a cool day. I hope that answers the question.
Greg.
Yeah, or send us an email and we might be able to point you in a direction or a local group that's closer to a site that you could collect from.
Bianca Gold's been putting the link to Biocontrol Hub in the chat. So if you'd like to use that to get to the information, that's a great resource. Are there any works in Victoria for oxeye daisy re-weevils? Also, does the infestation need to be dense in order for control to be effective?
Oxeye daisy research is underway in New South Wales, and so we certainly don't duplicate research projects. New South Wales is leading that research, Andrew McConaughey in particular, and so we'll maintain a watching brief. But certainly we share agents. Once New South Wales gets to the stage where they're releasing agents from quarantine, they would normally send starter colonies to other states, and we could rear them if we had the funding in Victoria. And then you would prioritise the sites. So initially we want to get the agents established, so we look for nice dense sites. But after that, they can be redistributed to other sites. It really just depends on your management priorities.
Excellent. Next question is, you mentioned it could be from 2 to 10 years to get a biocontrol agent approved. What biocontrol in the pipeline are you most excited for in the next few years? Great question.
Well, I'm most excited about
we will disagree
about
on
my
this.
Well, the most recent one that we have approved for release is a fruit feeding weevil for Sagittaria and Arrowhead, which are the aquatic weeds. So we're just waiting on the weevil, which is a Listerinotus species, to be added to the permitted import list, and then we will be hopefully ready to start releases. We've just also waiting for some funding. We've put in a grant application to the federal government. So yeah, that will be our next agent to go into our rearing and release pipeline. We've got a few others in the pipeline. Greg, do you want to talk about any of yours?
It'd be great to get a biocontrol agent out for silverleaf nightshade. It's such a difficult to control weed that's impacting pastures and cropping. And the priority is to find a very host-specific biocontrol agent, and that is difficult. The beetle that Ray showed on her slides was rejected, so I detected off-target damage. And so we rejected that agent. And because it's closely related to crops and many native species, we have to be really rigorous in our search. And we've actually gone back to Texas and to Argentina and searched for new insects that we think will be host specific. So that would be exciting for me.
And then there has also recently been the release of the Tradescantia fungus that CSIRO have been releasing in Victoria and other states as well. So that's also a fairly new biocontrol agent that is available for release.
Next question, how soon will silverleaf nightshade biocontrol be available?
I think I just answered that one, Kate. We rejected the biocontrol agent that we had in quarantine because we weren't satisfied that it was host specific enough. And we have two promising agents, one from Texas and one from Argentina. So they've commenced testing those and they appear to be host specific, and we'll probably import those, or at least one of those, in the next 6 months or so.
I've got a couple of comments to share with you, and also a reminder that both Ray and Greg are on Twitter. If you'd like to follow along their research, the links have been put in the chat, and they're probably on other socials as well, but I think Twitter's probably their main science outlet. So comments include, thanks Raelene for mentioning research on serrated tussock. The State Working Party welcome pending results. And woohoo for Arrowhead biocontrol. So some enthusiastic comments out there. Thank you both.
Fantastic.
Any more questions out there?
Another one that's come in, you just mentioned a fungus as a control. It's understanding that biocontrols usually use insects.
I don't know if it's a misunderstanding. It could just be that I talked a lot about insects because all of the biocontrol agents that we're currently working on are insects, but no, there are definitely fungi biocontrol agents that work very, very well. So yes, It's not just about insects. And there are also mites, and Greg was talking about the broom gall mite. Again, another very effective agent. The monitoring that we've been doing on the gall mite up at Dartmouth in the northeast of Victoria has shown that the mite, and also in combination with the rust, the broom rust fungus, is reducing the survival rate of broom. So broom plants that used to live over 25 years and now starting to die at just only 8 years of age. So that's a fantastic result for those two natural enemies of broom.
Another question, given that the Salvation Jane biocontrol is well established, how long would you expect that it will take to reduce the Jane that we are seeing at the moment?
Wow, that's a very good question. I think that if we continue with this rainfall for the next couple of years, and that allows those biocontrol agent populations to start increasing, hopefully, it still may take about 5 years. When you think, the thing with Patterson's curse is that the biocontrol agents are relatively slow to develop because they have only one generation per year. So that means that there's only one period of time that the adults are able to lay eggs, compared to, say, a mite, which might have multiple generations, lots of generations, a short generation time, and their numbers can build up. So yes, Patterson's curse can be a little bit slower. I would say give it a good 5 years for those insects to start building up, but they already are starting to build up. They've started to build up from about a year ago. So yes, we are definitely on the up. But that's not to say that if Patterson's curse is popping up in areas that it hasn't been before, and because of the movement of hay during the drought, you might be better off getting on top of the Patterson's curse now while you've got a chance.
All right. Another question, is there approval required for release of biocontrol agents on Crown land?
Greg, do you want to do this one?
Yeah, release of biocontrol agents on any lands requires the permission of the landowner or property manager in any case. It's the same with any weed management measure that you put in place. So often land care groups, for example, will work in conjunction with Parks Victoria's officers or their interstate equivalents. So there's usually a lot of collaboration that goes on ground. And once it's out in the field, biocontrol agents are a free management tool. And so often public land managers are more than willing to collaborate to release agents.
All right, a final comment, because I know we need to move on. Just a comment on the broom biocontrols. We had a fantastic result with the broom moth and rust, and had beautiful nursery site that we lost in all in the bushfires. The rust does work well in conjunction with the moth to keep on killing denuded plants over a 4-year period.
Yeah, it was a real shame about the bushfires destroying the few good sites that we have at that stage. And so release strategies often aim to conduct lots of releases in many different areas, but that has to be balanced with how many agents you release at any one site, because you want to maximise establishment. So there's often a trade-off. If you only have a few large releases, establishment can be pretty much guaranteed often. But if you have a very large fire, you could lose all of the sites, and that's what happened in that case, in which case you've got to start again and mass rear them and redistribute them. So often we'll make that trade-off. We'll have as many agents released as possible at a site, but we'll try and release them at many different sites across a very large area to safeguard against those extreme weather or fire conditions.
Thanks, Greg. Thank you so much, Kate, for doing that and for answering all the questions, Greg and Ray. It was fantastic. So Lee Mead is a volunteer and is the current president of the Tarrangower Cactus Control Group. Lee started working in veterinary science after completing a Bachelor of Science, and after a few years moved into medical research. She then completed her medical science in cancer genetics and worked in several hospitals and universities in Melbourne. However, being a country girl at heart, she had a midlife crisis, bought a post office in Malden, and had a tree change to central Victoria. Moving to the bush renewed her love for the Aussie nature, and she soon became a volunteer with the local land care groups. Lee retired 8 years ago and has since become a full-time weed warrior. Thanks, Lee. Over to you.
Thanks, Beck. Everyone hear me all right? Yeah.
Perfect.
Okay, as Beck said, I'm from the Tarrangower Cactus Control Group, and I'm going to tell you about our experience using the Cochineal insect as a biological control on a cactus species. So I'll tell you a little bit about the cactus problem that we have and our land care group, and then a little bit about the Cochineal biocontrol and our project that we've had running. So wheel cactus is an introduced species from Mexico and reproduces by both seed and vegetatively. It's mostly spread by birds eating the fruit, but it's also widely spread by water and rain and waterways. It's highly invasive and extremely difficult to kill. It's, as its name says, it's very robust. It's been a declared noxious weed in Victoria since the early 1960s, and it's a weed of national significance since 2012. This photo shows the typical infestations that we have around Malden in central Victoria in the Mount Alexander Shire. This Opuntioid species grows particularly well in granitic soils when we have lots of big outcrops, hilly outcrops of granite boulders. So we have lots of inaccessible landscapes like this where infestations have become extremely dense. And we also have locally we have a lot of smaller lifestyle properties, and so we have a lot of absentee landholders. This map just shows where we are in our area of concern. This is Bendigo up here and Castleman down here, and our group is based here in Malden. We've got two core infestations of wheel cactus just west of Malden. These are both on private properties, but they not only impact their neighbouring properties, but they also impact our Malden Historic Reserve, which we've got a lot of beautiful parklands surrounding the township of Malden. So our land care group formed in 2005. So for the last 15 years, we've been working on increasing community awareness about this particular species and the problem that we have and how invasive it is. We participate in lots of community events every year at the local agricultural show, and every year we enter in the Malden Easter Parade. Several information brochures over the years and distributed them in many different ways, including mail distribution, and we've produced farm fence signs and our t-shirts, but we've also do lots of media releases, and we have an informative website set up. Our group also tries to encourage the local landowners to participate in control management. So we hold about 6 or 8 community field days every year at different locations around the district. So, and our aim at our field days is to demonstrate the best control techniques. We provide incentives to anyone who wants to participate. We hand out these backpacks for injecting the plants and hose to manually dig. And at these field days, we try and promote networking between the neighbours. Our group has also trialled lots of different control techniques, of course, we tried spraying, but this particular species has got such a thick, waxy skin that it doesn't absorb spraying. So we've come to the conclusion that in our circumstance, chemical direct injecting herbicide into the lobes, into the large plants, and digging and burying the smaller plants are the best techniques that we can use. And we've got a couple of examples here of how successful these techniques can be. When we have conscientious landholders and they use the injecting and digging techniques, we can get very good results. It's time-consuming and laborious and costly, but it is possible. This is another example of how effective the techniques can be. If you've got access to the landscape, it's also quite possible to control. Once you've got rid of the really bad infestations, it's quite easy to keep on top of the regrowth with injecting and digging. However, we have always an ongoing problem with reinfestation because we have so many non-compliant landholders. This slide shows an example of the fence line between a non-compliant and a compliant neighbour. So we have continual reinfestation mostly from absentee landholder properties. And yes, this is another example of an absentee property where most of these owners, of course, are either ignorant or totally indifferent to their responsibilities to control their noxious weeds. And reinfestation is a problem too because we have lots of landscapes that are really difficult to deal with. The infestations become overwhelming for the landowners and they just can't deal with their infestations. So we decided that we needed, to try an additional tool, and we wanted to try biological control. Cochineal is the insect that is a biological control for cacti. This insect is indeed the little insect that produces cochineal dye for the food and clothing. It's a little scale insect, and the female insect attaches to the lobe of the cactus plant and sucks the sap out of the plant. This female covers itself with a protective layer of this white, waxy, fibrous stuff, and the males are much smaller and are mobile and crawl around. But nymphs of both genders are blown around by the wind, and this is how the insects are carried or spread between plants. Cochineal has been used in many countries to contain opuntioid cacti infestations, and as Ray talked about, it is a natural enemy of the cacti. It's also sourced from Mexico, and about 4 species of cochineal were introduced from Mexico in the early 1900s, mostly to Queensland, where they were testing to try and control the problems that they had with prickly pear and coral cactus. So the cochineal species have been in Australia for a long time, and South Australia started using cochineal for wheel cactus in 2000, in the century, and they've had quite good successes. This slide shows the ideal situation where the lobes are absolutely covered with the female insects that are sucking the sap. I was lucky enough to be in Spain a couple of years ago, and I did witness cochineal in action. So in lots of sort of areas, public parkland areas, there were opuntioid cacti with cochineal on them, and these 2 species have obviously reached equilibrium. there are several species of cochineal, and they are highly host specific. There are the cochineal species really only attack one or two different opuntioid species, and this table just is a small sample of some of the many opuntioid cacti species and the different or the particular cochineal species that will attack that particular opuntioid species. So the advantages of using cochineal insects for biocontrol, and the most obvious, as Greg said, it's cost effective because basically it's free. And using this as an additional tool reduces the amount of herbicide that we need to use, and which is a good thing because it seems that there are more and more objection to chemicals as time goes on. And this insect has the ability to kill small plants like this slide shows, and it has the ability to reduce fruit and seed production on the larger mature plants. Cochineal also, of course, reaches those inaccessible sites and locations, and most importantly, it blows across boundary fences to the non-compliant landholder areas. And if the insects populations are around, it can reduce the follow-up time that's required. This photo shows a wheel cactus plant that had been injected with herbicide, but invariably there is regrowth on these plants, and if we've got cochineal around, hopefully that will deal with the regrowth on those plants. Of course, all of these biocontrols and all of the insect species, etc., have limitations. As Ray said, most of them grow and populations take a very to get established. These cochineal species can take several years to reach a critical mass. And like was mentioned before, the biocontrol species are usually only effective in dense weed infestations, and this is the case with cochineal too. Cochineal can be impacted by weather, and not just bushfires, but the insects can be washed off the plants, and the insects definitely prefer hotter and drier climates. The insects can also be eaten by other insects if there's ants or beetles around. And as Ray said, the biocontrol doesn't ever eradicate the host. The two populations just end up in equilibrium, but it does help control. So our group was interested to trial these insects, and we obtained some samples from South Australia where they were using them, and we released them at a couple of local sites and waited for a few years. And it showed that they do survive in our local climate, and the population did increase over about a 3-year period. It definitely impacted small plants, and it's easy to harvest and redistribute. So we designed a project and applied for funding to try and introduce the new tool. And try and establish more integrated management plans with our local land managers and landowners. We were hoping that by introducing a cheap and easy new tool, that it will remotivate our landholders to participate in control, and of course hoping that this insect will help decrease the local seed bank and problems with reinfestations, and perhaps most importantly, try and create better, more sustainable long-term control strategies. Our project was funded by Landcare Victoria, and we did this work last year. First of all, we created a new brochure, information brochure about the insect to hand out, and held a few local community events to introduce this insect to our local community. And then we introduced the insects to 6 different properties. So here again, we're here in Malden, and our 2 core infestations are these 2 red squares. And we chose 3 properties that were nearby to each of those core infestations. So we introduced the Cochineal insects to those properties, and then created an integrated management plan to present to these landowners that incorporated to continue to inject and dig and spread these insects. As part of the project, we also built and established a Cochineal nursery. So we continue to propagate these insects in here, and these samples are available to anybody who wants to try and use this particular Cochineal species with Opuntia cacti. And since the project this year, we've seeded 4 more sites locally, and we've uploaded these sites onto the Biocontrol hub that Greg was just telling us about. So with some of our our insects from our nursery are already in action. Ray here passed on some of our Cochineal insects to Parks Victoria staff, and in collaboration with a local volunteer group, they've seeded these insects in the Chiltern Pilot National Park. And a month after they seeded these insects, they were already growing and spreading nicely. So that's a great story, and I think that is a good indication of these insects preferring a hotter and drier climate. As part of a follow-up for our project, we've formed the Cochineal subcommittee, and we're planning to follow up with each of the 6 properties every year. So far this year, we've just followed up with the first 2 properties, and sadly there hasn't been much action from either the insects or the owners. This photo shows, you know, there's a little bit of growth after 18 months at the first property. And we've set up photo points at each of the properties so that over the years, hopefully we will see some changes. But I'm sort of assuming that our very wet year is part of the problem as to why the insects haven't done much. And I'm sort of assuming that our difficult and challenging 2020 is part of the reason why the owners haven't done much. Anyway, but our key messages to our local landholders is that biocontrol is very slow to become established, and it's not the silver bullet. So all land managers need long-term management plans that they incorporate different tools. So we need to reinforce the necessity of using integrated management, using different tools. You need to manually assist the insects to help them spread quicker and the populations grow better. And everyone needs to continue to inject the mature fruiting plants that are still producing lots of seed. And if I take my, thanks Beck, last slide, if I take my Wilp cactus hat off for a moment and some closing comments as a volunteer, and as keen bushwalker, I see so many established weed species around our environment, and there appears to be little, if possibly no, control treatments year after year. And I think biological control agents can play a significant role as an additional tool in trying to help control these infestations. This paddock of Patterson's Curse is across the road from my house on Crown land, and there's blackberries and broom and bridal creeper and all sorts of weeds out there that just seem to be totally out of control. So I personally think that biological control agents need to be promoted and encouraged more, and at all levels, with private property owners, volunteers, and government land managers. And finally, I'd just like to thank all the supporters of our land care group, and thank you for listening.
Thank you so much, Lee. That was fantastic. Awesome to get a community perspective. Over to you, Kate.
A couple of questions for Lee. Fantastic presentation. Thank you. How deep do the small plants need to be buried?
Okay. They need to be well buried, probably half a metre of soil. But the one of the good features of wheel cactus is that they are very shallow rooted and they're very easy to dig up. So it is quite easy to dig the smaller plants, and it doesn't matter if you leave some root behind, they don't grow from the roots. So we do encourage landholders who have a lot of small plants to actually dig a pit on their property with a front end loader or something, and dump all of their small plants in there. Often they start decomposing if it rains a bit, but then they can bury them, and half a metre is a good depth.
Does department enforce compliance on cactus control?
Wouldn't we love that to happen? We have approached the state government a couple of times to try and start compliance on wheel cactus. We've basically been brushed off both times. The first time we were pretty much told that they don't have the resources, the staff or the resources to take on compliance of wheel cactus. The second time, we were told that they need to have lots of complaints for the same weed to have to start compliance. So we have been trying to encourage neighbours of the worst properties to complain, to send in a complaint, but we're finding it very difficult. People don't want to complain about their neighbours. They're very adverse to complaining about their neighbours. So we haven't been able to get any compliance happening on wheel cactus yet.
A number of comments on great presentation. Thank you, Lee. Loads of good work happening around Malden and the Tarrangower Cactus Control Group. Well done, and thanks for sharing, Lee. Great before and after pics, Lee. That's such a useful tool, isn't it, Lee?
Yeah.
And
a lot of work to do get that to happen, but anyway, they're great pics.
Really worthwhile. And you have a very cohesive, enthusiastic group, Lee. How do you maintain that enthusiasm for such a difficult challenge that you have?
That is a very good question, because it is a very difficult plant to kill, and we still have a huge problem even after the 15 years of tiresome work. I think we just have a core group of people who really are determined to try and get rid of this weed. It is such a horrible weed, and we've had lots of different volunteers over the years. You know, we sort of get 30 to 50 volunteers at our field days, but over the years they've changed a lot. So people come and go, but I think it is such a ghastly plant. And when you see the core infestations of this plant, it really people just, you know, are driven to try and get rid of it. It's it's, you know, it's quite addictive killing willow cactus too, so that helps.
We
feed them well. Sorry, Kate. We feed them well too.
You feed the participants, not the cactus.
Yes, sorry, we feed the volunteers well too. Yes,
I can vouch for that, definitely.
Also a reminder that there are loads of really good links in the chat in the speech bubble area of your screen. And one final question, can small cactus be disposed of at the council landfill, please?
We do have some. We receive funding so that we have free disposal of small cacti at our local mould and transfer station, and they go into the normal landfill, and in theory they will be buried with landfill. It's not ideal because there is a risk that they will escape, but in theory, yes. But it's just at our local. That free disposal is just at our local transferred station.
Thank you, Lee, and a reminder to everyone that there is the link to the survey. We'd love your feedback also in the chat. Thanks, Lee.
No problems.
Thank you so much, Lee. I just find the Tarrangower group just so inspiring. I love it. So thank you so much for giving that presentation. It was great. A big thanks to all our presenters, Greg, Ray and Lee. We really appreciate you giving us an overview of biocontrol, and I'm sure you can all agree we've all learnt something today. I know I have, at least anyway. So thank you to all our partners, the Weeds at the Early Stage of Invasion team. So Kate and Goldie, you've done an amazing job helping out today. We really appreciate it. To Connecting Country and Weed Society of Victoria, who helped promote this event, thank you very much. And we'd also like to thank Arthur Riley Institute for their support and assistance today for this Loddon Mallee Connecting Communities event. The Weed Society is looking to run some more online webinars as well, focused on weeds. So if there's any specific topics that you're interested in and you'd like us to cover, please let us know in the evaluation form that Goldie's posted in the chat. I'll also be sending it to you after this event as well. So we'd love to get what you think, because many of us, this is our first webinar. So let us know, you know, what you liked, what worked, and what things we could improve as well. We're always happy to get feedback. So yeah, so just make sure that you fill that in for us. That'd be great. So thanks again for taking the time to come along this morning and ask lots of great questions. We really appreciate your support. And watch out for an email from me in the next couple of days with the recording as well. So you can send that to anyone, and it'll also be put on the Swift website link as well. So we'll send that around. So that's the end of the formal proceedings for this morning for the Biocontrol event. We'll be commencing the Weed Society of Victoria's AGM at 11:30. So everyone is welcome to join. So we'll just take a quick 5-minute break, and we'll commence in 5 minutes. So thanks again for those who are heading off. Take care, and hopefully we'll see you soon for another event like this. Thank you.